We're going to go on with the rest of our program. I'm going to be making an introduction of Dr. Giulia Fragola. She obtained her PhD in 2014 at the European School of Molecular Medicine in Milan, Italy, characterizing the role of epigenetic markers in somatic cell reprogramming. I know you all know what that is. She then started her postdoctoral work, and I'm going to say worked in Mark Z's lab at the University of North Carolina of Chapel Hill, developing a CRSPR. I'm not sure what that is either. It's a base gene therapy for treatment in Angelman syndrome and studying the role of DNA damage in neurodegeneration. In 2020, Dr. Fragola started her research associate position in Dr. Todd Cohen's lab at the Department of Neurology at the University of North Carolina at Chapel Hill. Here she's applying her experience in gene therapy to develop treatments for Alzheimer's disease and study possible environmental contributors to the onset of neurodegeneration. So, Dr. Giulia Fragola, thank you for being here, and please come up and share your story.
Thank you so much. It's a pleasure to be here. Can you all hear me? I'm gonna shout a little bit. So, thank you for inviting me, and today I'm gonna talk about my project, which is about using tiny molecular scissors as a therapy for Alzheimer's disease. So let's first start by talking about AD or Alzheimer's disease. You are probably familiar with the most important symptoms of this disease, which is time confusion, memory loss, changes in personality, and energy loss, partly due to the fact that you also lose sleep. Unfortunately, in the US today, there are more than 6 million people that are affected by Alzheimer's disease, and this is supposed to double by 2060. So it's a very worrying scenario, but I believe we can do something to stop it. And the answer to this question is to look in the brain. So if we look in the brain of a AD patient and we compare it to a normal aging individual, you can definitely already tell that there is a shrinkage of the brain, meaning a lot of important regions are lost, which is the cause of the symptoms that the patient has. But there are two main things that every brain with AD has. If you look with a very big, very potent magnification glass, and these are the plaques and tangles. So these two are basically waste sites for two proteins that are highly produced during Alzheimer's disease. And so these are collected in this place, in these waste sites. And they are believed to be toxic and the cause of all the behavioral symptoms that are shown in AD. So researchers try to lower down these elements, these waste sites, and they started from A-beta plaques. So A-beta is the famous one of the two proteins that is overproduced in the patients. They managed to decrease the plaques, but most of the treatments that they developed failed to cause any benefits to the patients. So the level of dementia wasn't affected. Therefore, they then moved to look at tangles and try to start new therapies to lower these elements, these waste sites. And there are very promising data by target. So this is a very promising way to treat AD. So one of the major findings that led researchers to move to tangles is actually coming from mice. So yes, mice can get AD, and we can study Alzheimer's disease in mice, but how do we do that? So it's kind of similar to how people try to look for things in their house that they lost, or like finding their bedroom. So also mice can learn to find things. And the way we measure that is actually dropping them in a pool of water. This is called a water maze, and where there is a hidden platform in the pool. And mice just at the beginning, they will look around and swim until they find the platform, And then the next time you drop them in, they become better and faster at finding that platform. AD mice are not very good at that. In fact, they will just keep swimming and swimming and swimming, trying to find this platform, and they cannot memorize the way to go there. And it turns out that if you remove the tau gene, so if you remove these proteins from their brain, they actually become much better at doing that. So this is a very good sign that tau is is a perfect candidate for gene therapy. But how can we lower tau tangles, right? So it's kind of like dealing with plastic waste in the ocean. So tau are like these plastic bottles that gets accumulated in the ocean, and there are two main things that you can do. One thing is to clean it up, and so basically continue cleaning up, making sure that you are keeping up with the production. And today there are 9 therapies that are immunotherapies, which is what they do. They kind of clean up the tangles that are in clinical trial, and they are very promising. The second option is just to stop the production of tau. So do we really need all this plastic in the ocean? Like, do we really need to produce all this plastic? So today there is only one treatment that is in clinical trial. It's definitely safe and effective at lowering tau, but so far we don't have data on cognition, so whether this treatment is actually making patients feel better. There is only a small limitation of these. This treatment is not long-lasting, so patients need to go through several injections every 2 weeks or every month to actually keep the therapy working. And this is both economically, it's an economical burden, but also for the patient, it's not awesome because you need to do intrathecal injections, so every now and then. So we thought about another approach that is both safe and efficient, but also long-lasting to lower tau tangles. And so the very first step that was actually to, among all the tools that are available in gene therapy, to identify a safe and efficient way to stop tau production. So in real life, proteins, which is those elements that do basically the function that they're in our body, so the tau protein are produced from 2 main molecules, the messenger RNA and the DNA. So the DNA has basically the information to produce the protein. And while the RNA is like a messenger molecules that go outside from the nucleus where the DNA is stored and brings copies of this instruction to the chain machinery. So if we can stop the either production of the copy of the messenger RNA, or if we delete the information from the DNA, then we basically stop the production of tau. This can be done by using tiny molecular scissors, which are called CRISPR. You may have heard of these. So CRISPR is a tool that allows scientists to cut a specific site of the DNA and therefore get rid of genes that are either mutated or cause diseases. Although touching like the instruction, the main instruction, may be dangerous because even though it's a very efficient and very specific tool, it can also make errors. And so if you make errors in the main copy of your book, then you lose that information forever. So we thought about, instead of touching the DNA, to touch the messenger RNA, which is continuously produced. So it's just copying what is in the main manual. And we found another tool, which is called CRISPR-Cas13. It's not Cas9, which is touches the DNA. It's Cas13, which is able to cut the RNA molecules and get rid of those information. So how do you develop these, and how can you test actually that is effective? So we wanted to start easy and try to decrease tau in a dish before going to animals. So I told you that CRISPR is very specific. It can only cut the RNA that you design it to target. But how does it do that? So it turns out it does that through another RNA molecules, which is called a guide RNA, which perfectly fits to your target RNA, to tau. So the secret for a specific CRISPR therapy, an efficient CRISPR therapy, is to find the right guide RNA, so the right piece of the puzzle that can attach very efficiently to your target RNA. So how do we do that? We spend a lot of time in our lab testing several different guide RNAs to find the perfect ones that can be very efficient at lowering tau tangles. And how did we do that? By using cells that express a green tau, which is a tau protein that is fused to a green fluorescent protein, which is kind of similar to those glowing fish that you can now find in pet store. So you can visually see that tau is there, and when you delete it, when you knock it down, the cells becomes blue. So this is how cells look when they get rid of tau. You can see these green things are cells expressing tau, and if you don't put any guide RNA, you still have that. But if you put very good guide RNAs with CRISPR, you get rid of those green cells. But there are also guide RNA that are not very good at that. And so we, through a series of experiments, repeating these experiments many times, we finally found 3 guide RNAs that are very, very efficient. So we found a possible tool to target tau. We managed to decrease tau in a dish, and now we basically need to think about how to deliver this therapy to a brain. And so we wanted to identify a safe, but also, like I said before, a long-lasting strategy to deliver it. So in gene therapy, a very common tool that is used is the use of adeno-associated viruses. These are like tiny molecular carriers that can deliver any gene therapy to the body. So you can fill them up with your favorite gene therapy and then inject them in the patient. I know virus, they may seem scary. This is not the influenza virus or COVID-19. This is actually a very safe virus. It doesn't cause any symptom to people. It's very well tolerated. It does not multiply. That means that you cannot pass it to other people around you. It's not infectious. And it doesn't mutate the DNA of the host. It's very efficient at delivering therapies, and most importantly, is long-lasting. It can stay there for decades after a single injection. These information are coming for more than 20 years of clinical trials, more than 200 clinical trials, and on more than 53 different diseases. So it's very, very solid information. It's not like a new technology. So we managed to put our favorite guide RNAs with the CRISPR in the AAV particles. And we are now ready to the following step, which is finally deliver these viruses to the brain of AD mice and get rid of those plastic patches in their brain. And the following most obvious step is to test whether these mice can actually get better at finding that famous Eden platform in the pool. So this is still work in progress, but I'm very hopeful because so many people in the lab are working on these projects, and I had the chance to work with them. There are many students like Kyle, Jasmeet, and Spencer that I had the pleasure to mentor, and then also senior scientists like Shi, which is our virus expert, and Audra. And of course, nothing would have been possible without the help of Todd Cohen, my boss, which is an assistant professor at the Department of Neurology at UNC. But the most important thanks goes to you and for this honor that you give me basically to present my data and to do this research. And this wouldn't have been possible without all of you. And can we find a cure for dementia? I believe we can, and I hope I convince you that there is hope. And definitely research is the answer, but there is no research without resources. So it may seem small, but small projects like these can reach a big impact. It's like throwing a rock in the lake. They can help people like me to establish in the field of dementia, become independent, and start new projects. They can inspire younger people, younger researchers like the picture I show you, to work on dementia. And also they can help us collect preliminary data to basically ask for more funding. And so with this, I want to thank you all, and I will open it for questions if you have any. Thank you so much.
So have you tested the viruses on any humans yet? And if not, when do you plan to do that?
Yeah, so what happens in research is that you first start, like I said, in a dish. When you have enough data, then you go to the mouse, which we haven't done yet, but we are ready to do that. It takes a lot of resources to work with mice, but we manage now to get enough funding from the government to actually start that step. So when you then prove that in the mouse there is a benefit, then you work with primates first, non-human primates, usually. And after that step, you can start the phase 1 of the clinical trial, which is safety first. And this is what I was telling you about the other therapy that is in clinical trial. So they found out that it's safe, and then it can lower tau levels. But in the second phase of the clinical trial is when you actually look at the benefits, if there is any benefit of the therapy. So we are now at the very early stages, but this is how everything starts. So, yeah. Another hand.
How do the mice get the dementia or Alzheimer's?
Yeah, that's a good question. So we make them. We basically make them express the A beta protein, the one that is responsible for the plaques. So that is supposed to be the very first event that happens in Alzheimer's disease. So once you basically put the plaques in their brain, then they'll start a chain of events that leads them to Alzheimer's disease. Yeah. It's sad, but this is how it works. Yeah. Yeah.
What is the projected cost for research as far as for the treatment?
The cost for the next research?
Correct. What is the projected?
So usually grants are like NIH grants from the government. They are around $500,000, so like 5 years. It's not so. Yeah, basically, if you count like the salary of all the researchers that are involved and the cost to have the money, to keep the mice living in their cages and feed them and everything, and the cost to produce the viruses, it's close to that amount. But like this is where the government helps, and these are very strong data. So we already got 200,000 grants for going forward with this research, and the more data you have, the bigger grants you can apply for. Yeah.
I was going to say, what is the timeline from taking it from the mice to the primates to a?
Yeah, I would say around 5 years if everything works fine and it's promising, and we want to go to the next step. It takes about 5 years, and then you can start working with so we don't do any non-primate work at UNC, but there are other labs, for example in California, that there are very few places in the US that do that because it's a very expensive and ethically involved research. So you would start getting in contact with them, and they would do tests of another 2-3 years. So I would say, from 7 years from now, then you can start a clinical trial, which usually lasts around 5 years to phase 1, and then another 5 years for phase 2. So that's, But other things are ongoing. This is an additional therapy that may be better than others. But like everything that's in the clinical trial now is having very positive like results. So I'm very hopeful. Yes.
So just one more question. You were talking about removing the towels and the tangles, but you also mentioned that the brain was shrinking. Is there any chance, like once it's gone, the brain will stay where it's at,
Yeah.
and it just won't get worse?
That's definitely the idea. So to stop the degeneration. But so some of these therapy can be we can think about them as a preventative cure. So if there are early signs, or in general, if like for example, other ways to image the amyloid much earlier than the symptoms occurs. So if there is anything that we can like diagnose before, we could just start those treatments to prevent further, like basically the degeneration of the brain. They cannot bring a very severe case back to normal, but they can prevent further degeneration.
Since all this with gene therapy is Alzheimer's entirely or largely genetic? You're genetically predisposed and it's not an outside thing? Is that where the research is?
No, so very like a minority of cases is caused by a single gene mutation, which is in the beta amyloid famous plaque forming protein. There's definitely genetic factors that could put you at risk, but like the vast majority of AD cases cannot be explained only by genes. And this is my other part of research. I'm studying environmental contributors to neurodegeneration. So it's kind of a mixture between genes, environment, and age, because age definitely brings up the chances of having Alzheimer's disease. And yeah, we are studying those too. But like we know down the pathway there are some proteins that are involved in the real toxicity of Alzheimer's disease, like tau. So you can just get rid of those to avoid further degeneration.
Did I see another hand?
Yeah.
Is there anything that you give to the mice to enable them to thoroughly accept the CRISPR? Any medication, anything that you do to the mice to allow them to accept the molecular scissor any better?
You mean if there is any rejection, like
Yes.
we would do with a transplant? So let's say if you inject them basically intrathecally, they will directly go to the brain, and they are not really rejected because it's like getting infected by a virus. And viruses are very good at infecting people. Now there are some situations in which, and they found this in some cases, rare cases, in which you already been infected with adenoviruses, for example, in your life, because these are viruses that are around, and you may have developed some immunity to that. And so they are working around some molecules that are outside the virus to change them so that they are not recognized by our immune system. And this is in development. Another thing interesting of these viruses is that they have the potential to be injected not like intrathecally, but just in the bloodstream and reach the brain. And so that's an advantage, but it works better with younger patients compared to older patients. But they are working again on these envelope proteins, they call them, so they are outside the virus to make it more efficient to go in the brain and stay there. Yeah.
Giulia,
Thank you so much.
thank you.
That was great.
So, thank you.